Hi friends, so we have completed our chapter Biotechnology Principle and Processes, and now we are moving on to the new chapter. The name of the chapter is Biotechnology and Its Application. Again, a very interesting chapter, but if you cannot understand the concept, it becomes difficult. So listen carefully, right? Now, the chapter is Biotechnology and Its Application. So, in this chapter, we are going to learn about the different applications, uses, significance, importance of biotechnology in different fields. Okay. So, as such, when you are saying this, biotechnology is used in innumerable number of fields, isn't it? Like it is used for therapy. Therapy means what? For treating diseases, for diagnosing diseases in the medical field, in research, for forensic studies we are using it, for making many products and industries we are using biotechnology. We are making many plants and many animals by rDNA technology. Different animals, different varieties of plants and animals are created with the help of what? This biotechnology. We are using this biotechnology in making many microorganisms which are helping in treating waste treatment, you know. So, in order to tackle waste, many microorganisms we are making and we are trying to use it for degradation of waste. In order to destroy the waste, we are using all these microorganisms, right. So, these microorganisms with the help of this biotechnology, rDNA technology only, we are making it. And another term what has been given in your book is bioremediation. So, what is bioremediation? Bioremediation, remedy means, you know, remedy means what? A thing in order to overcome that what you do, right. So, that is we call it as its remedy. Now, bioremediation. So, what is bioremediation? So, if there is a problem, I am using a biological source. So, it can be a living organism. Like for example, a tanker was carrying oil from one place to another. So, in the sea, all of a sudden there was a leakage in the tanker because of some reason, and there was oil spill. Now, this oil spill has to be overcome. We cannot you can imagine tanker, so much amount of oil will be there, isn't it? So, you can find around meters, the whole sea is surrounded by what? Oil now. Now, how to overcome this problem? Because if this oil spill is there, you can find the birds, the ecosystem, means the micro ecosystem which is present over there, it will be getting disturbed, right. So, in order to prevent that, what I am doing is that we are going to make some organism and put it in the ocean. That organism has the ability to digest this oil. Like for example, Pseudomonas aeruginosa is an organism which I am making it with the help of this biotechnology, and this organism, when I am putting it in the water, for them the food is oil. So, what they will do? They will eat up this oil, they will break down this oil, and the oil spill will be removed over that place. So, here when you are seeing the remedy is a biological organism, the microorganism which I am making it with the help of biotechnology. Okay. So, that is again a field of what? Bioremediation. That is why we use the term what? Bioremediation. As well as biotechnology, many organisms are being made for production of energy, for making from the waste to generate energy, biomass, like huge amount of energy can also be made with the help of what? This microorganisms which are engineered. It is not the natural one which is available, we are making them, right. So, these are all different fields of what? This biotechnology application. So, all these places you can find the biotechnology is playing a major role. Another one thing what has been mentioned in the introductory part is that there are three critical research areas of biotechnology. Okay. So, the first research area is providing best catalyst in the form of improved organism or as enzymes. So, biotechnology, with the help of biotechnology we are making what? Like for I told you in the last class, we are making insulin, we are making enzyme lipase, we are making things, okay, we are making many proteinaceous things, even we are making organisms which is not their natural form. I have introduced some foreign DNA into it, and this organism is different from the original one. Okay. So, we are making many organisms or we are making many products with the help of what? This biotechnology, and this is one research area. Second is that we are creating all the conditions which are favorable for making these organisms. So I want to produce some organism, a new organism, or I want to make organism which will be producing some enzymes or catalyst. For them, in the biotechnology, we are also dealing with creating a good atmosphere. The organism, just like that, they cannot grow. So for that, I have to create some critical conditions, critical atmosphere. So that setting of the atmosphere is also another field which is dealing with what? Biotechnology. Okay. So yet another field is creating optimum conditions in order to facilitate the growth of these organisms so that they can make their own products, and these products can be useful for mankind. Okay. And the last one is your another one research area regarding biotechnology is your downstream processing. We have already learned, isn't it, bioreactors. In spite of making all these products, we have to see to it that these products are safe. These products are not causing any problem. This product are, if it is consumable, whether it has undergone all clinical trials properly and whether it is pure, safe, then only we can get it into the market. So for purifying all these things, that process we call it as what? Downstream processing. What and all technologies are involved? That is also coming under what? Biotechnology. So these are the three critical research areas which are dealing with what? Applications of biotechnology, right? Now we are moving into the chapter. So this was only an introductory part. Most questions will not come from this part, but now we are moving on to the chapter, starting with biotechnology application in agriculture. Now we are going to see what is the application of biotechnology in agriculture. Okay, in this we will be learning two. One is your BT cotton, BT plant, and second one is your pest resistant variety, developing pest resistant variety. So these are the two topics what we are going to discuss in detail in biotechnology applications in agriculture. Before that, we need to know something regarding biotechnology. What is the role in agriculture, right? So the first one is that biotechnology for increasing food production. When you are seeing agriculture, it has to increase food production. Three main areas, three options are available in order to increase the food production, right? So I want to get good yield. For getting these good yield, I am having three options in front of me. Imagine I am a farmer. So I am having three options in front of me. What are the three options? Number one is agrochemical based agriculture. Agrochemical based agriculture means what? I am using chemicals like pesticides, fertilizers are being used. These are put and their yield is made much with the help of all these chemicals. So that is the first way by which I can improve. I can increase my yield, but it is not good. I know in the long run the soil will turn bad and it will be turned into a wasteland, not that good. Second is that organic agriculture. What is organic agriculture? I can improve the food variety, I can increase the quantity of food by going for organic agriculture. Organic means what? I am using all bio pesticides, bio fertilizers, manure. Okay, I am not going to harm any plants, animals, insects there. I am using all biological things which is safe. Okay, but this biological things, when you are seeing, it's a bit costly. At the same time, you can find properly they have their demerits. Okay, you cannot find that they are giving much yield in the long run, much amount of yield will be produced. No, to some extent, if it is a small scale, then this organic farming is good. So that is the second option. Like that also we can increase the yield, right? So first is using chemicals I can increase. Second is I can go for organic agriculture, and then I can go for what? Increasing my food production. Third is the best option nowadays. We are making genetically engineered crop, GMOs. What do we call them? We call them as GMOs. Means genetically modified organisms. I am making organism with the help of what technology? rDNA technology. I am making my organism. Like we have already discussed. In green revolution, what happened? The food quantity increased. But when you are seeing this green revolution, when it was done, they used hybrids. Hybrids like what? Pollen grains, the artificial hybridization we learned, isn't it? Pollen grains from the high yielding plant were dusted over the stigma of the disease resistant plant, and these two made to fertilize, and the seed which was produced was both high yielding as well as disease resistant. But this is a time consuming process, and most of the time sometimes it will not be giving good results. So that became very difficult. These hybrid, they need much amount of fertilizers, they need much amount of pesticides, then only what they started. They yield was much higher in the green revolution. So in order to get rid of these problems, okay, I want high yield. How? Because you know the population is going on increasing, the demand is more but the supply is less. In order to fulfill the demand of the country, in order to fulfill the demand of the people, we need what? We need much amount of food. So in order to get that one, I cannot go for trials and all. I want much amount of food is being produced. So for that, in which areas, what were the defects? That defects, to overcome that gene was introduced into those plants and they were made resistant to those varieties. Like for example, in one area the plant was not growing because there the region was drought, the water availability is less. In that case, I am introducing the gene which is resistant to drought. So automatically the plant will now go there, yes or no? Yes. In one place there was much amount of pest, because of which the plant was not growing. Now what I have done, I have introduced a gene which is responsible for resistant to pest. Now if that plant is grown there, whether they will grow or not? Yes, because I have introduced a gene which is resistant to pest. So like that, I found out the problem with the plants, and that problem was solved by taking out the undesirable characters and putting in the desirable characters by cutting and pasting. That process we call it as what? rDNA technology. With that, I made plants. So these plants which I made for making different products, right? Like maybe it is a fiber for making cotton, or it can be vegetables, whatever. These are made by me. I am making it by cutting some segments, introducing some other segments into it. So this sort of plants, what I am making it, we call it as tailor-made plants. What do we call them? We call them as tailor-made plants, because I am the tailor. I am cutting and stitching it, and I am making a dress out of it, means a plant out of it. So in that case, I call this one as what? Tailor-made plants are being made. And these plants, these organisms which are made like this with the help of rDNA technology, we call it as what? GMOs. So what is GMOs? It is genetically modified organism. What is GMOs? It is genetically modified organisms, right? Yes. So with this genetically modified organism, what are the benefits? Okay, genetically modified organism, what benefits they have? You see, first is that with the help of genetically modified organisms, means we can make genetically modified organisms, and these organisms are first tolerant to abiotic stresses. What do you mean by tolerant to abiotic stresses? Abiotic means, you know, non-living factors. So what can be the non-living factors? It can be like your cold, it can be drought, it can be salt, it can be heat. So the genetically modified organism which we are making, it is tolerant to, cold, means in place where it is too cold also they can grow. It is tolerant to drought, another abiotic stress, means if the water availability is less also they can grow, no problem. Another one is that it is tolerant to salt. If the soil is salty also, no problem, I will grow. Like that plants I am making, okay. And the last one is, you know, global warming. So the temperature is rising, the tolerant to heat. So the plant is now I am making which is tolerant to heat. So in that heat means the hot regions also they can grow. So like this, these genetically modified organisms are having the capability to live in these abiotic stresses, conditions where there are much amount of stress caused due to abiotic factors, right. Next is that reduce reliance on chemical pesticides. Why? Because I am making a pest resistant plant. So inside the plant itself there is a toxin, which is not allowing the pests to come and attack. So if the plant itself is having a toxin which is not going to allow the pest to come near it. Okay, so I am making it. I am introducing a gene which is resistant to pest. Now this resistant to pest gene will code for a protein. By seeing that protein, smelling that protein itself, the pest will not try to go near it. Okay. So in that case, I am making a pest resistant plant. So if I am making a pest resistant plant, do I need to apply chemically pesticides outside? No, already the plant is pest resistant. So there is no use of pesticides. Chemical pesticides are not needed when I am developing this genetically modified organism. Next is that they also help to reduce post-harvest losses. Very important, listen carefully. What do you mean by post-harvest losses? Means after harvesting. Harvesting means the food has been taken. Thereafter, till it has been supplied to the market and we are consuming it, all these time period is there now. From the harvesting till it is going to the market and coming to the consumers, all these things are post-harvest losses. Okay. Now, most of the post-harvest losses is taking place because you see, previously the vegetables, whatever we buy, we people are like that. We think that if the vegetable is good looking, there is no impressions, there is no color discolorations, all these things means the vegetable is good. I will buy the one which is very soothing to my eyes to see, very nice. So that one only I will be buying. And I will be neglecting which one? The one which is not that good, shape is not proper, discoloration is there, but maybe that is the one which is organic, it's good. And the other one, when you are seeing, they have applied all these chemical fertilizers, waxing and all, and have done, and it is bad. In spite of that, we will be going for what? The one which is soothing to our eyes, isn't it? So in the post-harvest cases, what happens is that when they are making these vegetables, fruits, everything, because of not having soothing to the eyes, when you are seeing they are having some discoloration, all those factors, we people started neglecting it. So like that, much amount of food which are produced are wasted because we don't go for that. We say no, usko hatao, you just give me the one which is very good. I don't want the one which is not looking that good. So in that case, what happened? Much amount of food is wasted. So with the help of this genetically modified organisms, we are seeing to it that the crops which are being cultivated are having the appearance which is somewhat soothing, so that nobody will say that no, because it will be having good color, good shape, everything, so that all the things which are produced will have these characteristics, so that the food will not go waste. Okay. So this one we call it as what? Post-harvest losses can be tackled by making this genetically modified organisms. Next is, this genetically modified organisms are having, I am introducing gene which are utilizing the minerals in a proper way. They are not over-exploiting. Over-exploiting means what? Simply taking it, they are not using it, simply getting stored somewhere in the plants and it is wasted. No, it's not like that. So these genetically modified organisms, we are introducing genes which will increase the efficiency of what? The mineral usage. The minerals which are taken from the soil are used in a proper way. Over exploitation of minerals not taking place, and how much is wanted, that much only they are taking, much they are not taking. So what will happen? I need not apply more fertilizers, isn't it? If the minerals are taken properly and used properly, and over they are not taking from the soil, then the soil will have some minerals there. Next time also, when I am going to sow the seeds, the minerals are already available, I need not add any fertilizers. So like that also, these genetically modified organism is playing a major role. And with the help of biotechnology, I am making an organisms which are having genes which are coding for specific nutrients. Like for example, golden rice is the rice which is vitamin A enriched rice, which means that this rice contains huge amount of vitamin A. Why? Because I have introduced a gene which is coding for vitamin A, and this rice will be having what? More amount of vitamin A. You know, for eye vision, this vitamin A is good. So in order to those people are having this problem with some diseases, so that particular mineral nutrient or vitamins are introduced, the genes which are coding for that are introduced into these plants, and these plants are made available with that nutrient. So, if you are having that as a food, you are getting the nutrient in your body. So, like that also we are making what? Genetically modified organisms. These are all the good sides of what? Genetically modified organisms. Obviously, there will be bad effects also, but now we are going to see only the good effects. Okay. So, genetically modified organisms are the full form for what? GMO. So, the GMO is having all these good characteristics for which mostly we are going for what? This GMOs nowadays, right? That's why they have increased the yield and we are getting food. If these GMOs are not available, hybrid varieties are not available, then we would be starving, because the natural varieties do not grow much, as well as you can find the production is very less, right? Now, applications in the field of agriculture, as I told you, we will be discussing two topics. One is the BT cotton and second one is pest resistant tobacco. Okay. So, two things we are going to discuss. One is what? The BT cotton, and the second one is pest resistant tobacco. So, the first one what we are going to discuss is BT cotton, right? So, moving on to BT cotton. Listen very carefully, a bit understanding one concept, so you have to understand. So, see to it that you are listening properly, right? So, everyone is fine listening, right? Fine. So, BT. So, what is BT standing for? BT stands for a bacteria. The name of the bacteria is Bacillus thuringiensis, Bacillus genus, thuringiensis species. So, what is BT? BT is standing for a name of a bacteria. What is the name of the bacteria? Bacillus thuringiensis. Okay. In this bacteria, there is a gene. The name of the genus, that particular segment of gene, is called as the BT gene. What is the name of the gene? BT gene. Okay. So, this BT gene, when you are seeing, it is otherwise called as cry gene. What do we call them? It can also be called as what? It is can also be called as cry gene. Why? Listen carefully. So, Bacillus thuringiensis is the bacteria. In that bacteria, in the nuclear, you can find the DNA, there is a gene. What is the name of the gene? BT gene. And this BT gene can also be called as what? Cry gene. Why it is called as cry gene? Because it is going to give rise, this gene is coding for a protein. What it is going to coding for? It will helping in the production of a protein called as Cryoprotein. What is the protein's name? Cryoprotein. So, what is this Cryoprotein? This Cryoprotein is being given a name like this because they are producing crystalline material. They are producing what? A crystalline structure of protein. That's why the name is what? Cryoprotein. Cry stands for crystalline, right? So, BT is Bacillus thuringiensis, a bacteria. Inside the bacteria there is a DNA. In the DNA there is a gene called as BT gene, and this BT gene can also be called as cry gene, and this BT gene is helping in the production of a protein called as what? Cryoprotein, and this cryoprotein is usually not active, it is inactive form. This crystal is an inactive form. Till this you have understood, right? Now listen, this BT gene is producing cryoprotein, right? For producing this protein from this gene, there is a process involved. We call it as transcription and translation. What is that process called? We call them as transcription and translation. Okay, in that, to produce a protein from DNA, I need two processes. One is transcription, second one is translation. Okay. Now we are going to discuss about what is transcription and what is translation. Not in detail, because it will be dealt in the chapter 6, the protein synthesis. You will be learning in detail about what? The transcription and translation. But here I have to say transcription and translation, then only you will understand the concept. A small in gist I will tell what is transcription and what is translation. Okay. In chapter 6 you will be learning in detail. Now moving on to transcription translation. Listen very carefully. With the help of a story we can explain what is this transcription and what is translation. See, I love my mother making chili gobi. Okay, my mother used to make very delicious chili gobi. I love to eat that chili gobi. So what happened is that for my studies I went out. In that area, okay, chili gobi you can avail anywhere, isn't it? I was getting chili gobi, but that chili gobi was not that delicious like my mother's recipe, and I was tempted to eat the chili gobi what my mother makes. Now what happened? I want my mother's chili gobi. So sitting far away, I cannot have it. So one idea struck me. I can go and ask my mother over phone how to make this chili gobi, okay? Otherwise I will go to my mother this time and I will ask her how to make chili gobi, right? So what my mother did is that she was very busy, so she gave me a book. What she gave? She gave me a book. So that was a book which consisted of the recipes what she used to make. With the help of that book, all the ingredients and how much amount, everything is being given in that book. So in that only you can also find the recipe for what? The chili gobi. So I took that book. So that book, from that I went on to the page. Which one? The chili gobi page. Thereafter I read all the things, how the procedure is, and from that I made what? The chili gobi, right? So this is what I did, and when I tasted, it was just the taste as my mother had made it, right? So the same thing goes with the DNA also for making protein. So DNA, when you see, it's a big thing where you can find innumerable number of genes. Like a book which contains many pages, the DNA contains many genes which are coding for many things, like many recipes, okay? So out of that, only one particular gene is going to give me that food, isn't it? That protein, that is the chili gobi to make. So for that, what I have to do, from this DNA, the whole book I cannot take and go, right? Because my mother needs it, yeah? But I want only the chili gobi. What I will do? I will copy the page where the chili gobi is given. So that one I will be just noting it down in a bit of paper, right? So from the DNA, this copying it down which is being done, that process we call it as transcription. What is that process? Transcription. So DNA, from that particular gene which is coding for one particular protein is being copied in the form, a page, right? So that is RNA. What is that? RNA. So DNA is now copied and it is converted into what? It is converted into RNA. The RNA are of three types: mRNA, tRNA, rRNA. Here it is mRNA, messenger RNA. The message is being copied, the recipe is being copied, okay? So that one we call it as what? Transcription. So what is transcription? DNA to mRNA is called as what? Transcription. So DNA is the whole lot. From that one particular gene which is going to produce my protein, that is being copied it down, okay? It's not made, it's just copied. So that copying process we call it as what? Transcription. DNA to mRNA we call it as what? We call them as transcription. Now after it's been copied, I am doing everything and I am making the protein, right? So that process we call it as what? Translation. What is that process called? Translation. So in order to make any protein in your body, right, whether it is a hormone, whether it is an enzyme, all are proteins, right? So all these proteins to make, who is responsible? The DNA, the gene which is present in the DNA is responsible. So from that DNA, when we are seeing that this protein is needed, DNA will form what? mRNA. So in this DNA, there will be a segment will be copied from the DNA, the gene segment. That copying is done by whom? The messenger RNA. And this copied things is translated into what? The protein. So I am getting the protein. So in order to make a protein, how many processes are? Two processes. What are the two processes? One is transcription and another one is translation. With the help of transcription, I am getting mRNA from DNA, okay? Particular gene sequence is being copied. From that gene sequence, the processing is done, and what is happening? The translation, that is the protein is synthesized. Okay. So, for making any protein, whether it is enzyme, whether it is hormones, or whether it is any parts, we need what? We need the mRNA. So, the two processes are there, one is transcription and another one is translation. So, DNA to mRNA is transcription, mRNA to protein is translation, and this is a common procedure for making any type of protein. Okay. So, we give the term central dogma of protein synthesis. We call them as, for this equation, as central dogma of protein synthesis, wherein you can find the DNA, that is from the book, the recipe is being copied, the gene is being taken, okay, copied. So, this process we call it as what? Transcription. So, this process is transcription, DNA to RNA. Then RNA to protein, we call it as what? The translation. The food is being made, the protein is ready, right? Now, we will move into BT. Yes. So, now listen. So, what happened is that BT is a gene which is present in the DNA. The gene, from there what is produced? mRNA is produced, isn't it? And this mRNA is producing what? The protein translation. So, two processes is over here. So, what are the two processes? One is transcription and another one is translation, and the cryoprotein is formed. Okay. So, for making any protein, you have two processes, transcription and translation. From DNA to protein, you have two processes in between, transcription and translation. Now, listen very carefully. Now, this BT gene, which is produced by, which is present where? In Bacillus thuringiensis, this BT gene is isolated. I can isolate. Restriction enzyme I am adding, cutting into fragments, gel electrophoresis, BT gene is isolated. This BT gene is taken and it is introduced into the plant. Where it is introduced? Into the plant. So, it has gone into the plant now, plant cell. Okay. Now, in the plant cell, since the gene is introduced, whenever copies are produced, you can find the gene will be expressing, isn't it? So, when the gene is getting expressed, means what? It is going to produce the protein. That gene, BT gene, will produce what? The BT protein, isn't it? Cryoprotein they are going to produce. So, the plant, inside the plant, you can find this gene will be undergoing transcription, will be undergoing translation, and in the plant what will be produced? This protein will be produced and is stored. Right. This protein, I told you, it is crystalline protein, crystal-like structures which are stored. It can be stored in any part of the plant, like for example, it is stored in the leaf, right. So, here I have informed what it is stored in the leaf, there it is produced and stored. In every part it is stored, along with that in the leaf also this cryoprotein is present. I repeat, so from the Bacillus thuringiensis, I have taken out the BT gene, that BT gene is introduced into a plant. In that plant, what is taking place? The BT gene is doing its function. What is its function? To produce the protein, cryoprotein. So, transcription, translation is taking place and the cryoprotein is formed. Now, listen very carefully. This protein is present in the leaf now, everywhere in the plant. This protein, I told you, this cryoprotein is inactive. Inactive means what? It is not functional. But it can become toxin. When it can become toxin? When the medium turns alkaline, it can become toxin. When the medium turns alkaline, it can become toxin. In plant, there is no alkaline medium, so no problem, they are safe there. This toxin which is present in the plant is not going to cause any harm, because it is not affecting the plant. Plant is not getting affected, right. Now, what happens? Listen very carefully. Larva of pest, isn't it there? So, the larvas of the pest, like pest, you have 4 stages. Mostly these pest metamorphosis, they have egg, larva, pupa, adult. One caterpillar larva is coming and eating the leaf. Which leaf? That cryoprotein, the plant in which the BT present, that leaf they are eating. Now, what will go into their body? The protein will get into their body? Yes, it has gone into their body. So, in the larva, the protein has gone. After entering into their body, it is going to the gut intestine. So, there epithelial cells are present, right. And when you are seeing in the gut, the medium is alkaline. What is the medium in gut? It is alkaline. Now, this cryoprotein which was inactive, sleeping, right, has become what? It has become active. So now they are becoming active in the alkaline medium and it is turned into a toxin. What they will do, this cryoprotein, they will go and attach to the epithelial cells, to which they are going and attaching to the epithelial cells, and they are making pores. Yes, holes they are making. Now water will get into the cells will swell up, the cells will burst, the cells will rupture. Now when the epithelial cells gut of the larva is rupturing, so what is happening? The larva is killed. Imagine, many larvas are there, all are trying to go and eat. When they are eating, the toxin is going into their body, the cryoprotein. Thereafter, they are getting activated when they are reaching the gut because it is alkaline medium. So there they are getting killed. Now, if the larva is killed, do you think the next stage will go? Pupa? No. Next, adult? No. If the adult is not there, whether the reproduction is going to take place? No. So at last what will happen in the field? You cannot find this pest available because the pest is eradicated with the help of what? This BT, right? So this process is one method in developing what? BT varieties in agriculture with the help of what? BT gene, which I am engineering with the help of rDNA technology, introducing BT gene into the plant, thereby making the plant resistant to pest, right? So BT is one. So a bit of explanation. Some strains of Bacillus thuringiensis produce protein that can kill certain insects. Okay, they are toxic to certain. Some substances are toxic to plants, or some substances are toxic to some pests. That substance may not be toxic to us. If it is toxic to us, obviously we will not use it. If it is edible one, we will not be using it, because we feed, we are also having alkaline medium, acidic medium, we will be killed, isn't it? So they are testing it beforehand, okay, how much it is, concentration is less or not, whether it will affect humans or not, only that much they will be taking, okay. Yeah, so kill certain insects such as tobacco, budworm, arbworm, cleopterans, dipterans, all these can be killed. Means these larvas, they start to eat these plant, means they will be killed, right. So Bacillus thuringiensis form protein crystal, I told you, cryoprotein, during a particular phase of their growth. These crystals contain a toxic insecticidal protein, so it will attack the insects only. BT toxin protein exists as inactive protoxin, I told you, it exists in the inactive form, but once the insect ingested, inactive toxin is converted into active when it reaches the alkaline pH of the gut, which dissolve this crystal and it will become what? It will become toxin. Now, the activated toxin binds to the surface of the mid-gut epithelial cells. So, gut's epithelial cell is going and binding, and they are creating pores. The cells are swelling up because of intake of water. Cells will burst after swelling, cause the death of the insect. Specific BT toxin genes were isolated from Bacillus thuringiensis and incorporated into several crop plants such as cotton. So many plants, soybean, I told you, cabbage, like that they are introducing in order to get what? Insect resistant varieties, right. So the toxin is coded by a gene named Cry. So name of the toxin is what? Cryotoxin, okay, Cry gene. So different Cry genes are helping in controlling different insects, okay. Like for example, Cry1AC and Cry2AB, they are means that genes, when you are saying, they are coding for controlling whom? The cotton bollworms. So the cotton bollworms can be controlled by these two genes. What are the two genes? Cry1AC and Cry2AB. It is a different sequence, different gene, right, which is helping in what? Controlling bollworm. This gene, if it is introduced, bollworm will not grow there, they will be killed. And if I am introducing Cry1AB, okay, into the leaf, like in corn plant and all, if I am introducing, then when you are seeing, they are resistant to what? Means they will kill whom? The corn borer will be killed, okay. So like this you can find this insecticides plants can be developed with the help of what? This BT. This is the diagram depicting BT cotton. In the next class we will be discussing about the second application in agriculture, that is pest resistant tobacco variety. Hope till this what we have discussed is clear. Thank you.